Membrane receptors, as I told you, are present on the cell membrane. They are actually the transmembrane proteins embedded in such a way that the active site or binding site is located outside the cell. Due to this, they usually consist of three parts: the extracellular, the membrane spanning, and the intracellular domains. The extracellular N-terminal domain binds to the hormone and also has the glycosylation site. The extracellular domain that binds the receptor is often rich in cysteine residues, which form rigid pockets in which the hormone is bound. The transmembrane region consists of one or more segments made up of hydrophobic amino acids, arranged helically, whose role may include the anchoring of the receptor in the membrane. Disulfide linkages are present in between that make the structure stable in the membrane. The intracellular domain is the effector region of the receptor, which may be linked with another membrane protein system to perform biochemical changes. Three types of membrane proteins can be present with the intracellular domain: ligand-gated ion channels, G proteins, and kinase channels. Hence, receptors are classified as channel-linked receptors, G-protein coupled receptors, and kinase-linked receptors. Let's get into the details of each. Channel-linked receptors are also known as the ligand-gated ion channels or inotropic receptors. Receptors are linked with a group of proteins known as ion channels. When hormone binds to the receptor, it activates ion channels. Opening of ion channels allow movements of ion across them, such as sodium, potassium, and calcium ions. They pass through these ion channels. This passage of ion leads to changes in the electrical potential of the cell. It can cause either depolarization or hyperpolarization. Depolarization means less negative charge inside the cell. In other words, outward movements of negative ions or inward movements of the positive ions. And hyperpolarization is opposite. There are high negative charges inside the cells. In other words, outward movements of the positive ion or inward movements of the negative ions. These types of the receptors are usually present in the electrically active cells such as neurons. When a presynaptic neuron is excited, it releases a neurotransmitter, say acetylcholine, from the vesicle into the synaptic cleft. The neurotransmitters then binds to the receptors located on the postsynaptic neuron. If these receptors are ligand-gated ion channels, a resulting conformational change will open the ion channels, which leads to a flow of ion across the cell membrane. This in turn results in either a depolarization for an excitatory receptor response or a hyperpolarization for an inhibitory response. Now moving on to G-protein linked receptors. Other names are G-protein coupled receptors, GPCR, 7-pass transmembrane domain receptors, 7-TM receptor, serpentine receptors, or heptahelical receptors. Well, most of these names are related to their structures, as they pass through the cell membrane 7 times, or you can say that they have 7 alpha helix in the transmembrane part that spans the membrane. Looking deep into the structure of a G protein coupled receptor, it has an extracellular domain to which the hormone binds, and an intracellular domain linked with the G proteins. A G protein have three main parts: alpha subunit, beta subunit, and a gamma subunit. When activated, first a subunit substitutes a GDP molecule for a GTP molecule. This results in activation of G proteins. They can be either stimulatory or inhibitory. That is, they can cause an increased level of enzyme activity or decreased level of activity in the second messenger system. Now, what is the second messenger system? G proteins act as the first carrier of the signal from the hormone. They transmit this signal to the second carrier called the second messenger. Second messengers are small molecules that carry the messages of first messengers to the target. There are a number of such messengers, but two of them are more common: adenylate cyclase system and inositol triphosphate system. Let's get into the details of both. Now, activated subunit of G protein will bind to adenylate cyclase enzyme that is present in the cell membrane. Now, this binding will activate the adenylate cyclase. Activated adenylate cyclase will ultimately form cyclic adenosine monophosphate from adenosine triphosphate. Once formed in the cytoplasm, CAMP activates the catalytic subunit of specific protein kinase pKa, which forms part of a cascade of intracellular phosphorylation resulting in the cellular response. Since just one molecule of hormone can result in production of many molecules of CAMP, this is a very efficient means of amplifying the receptor. Receptor-hormone interaction. Once formed, CAMP is rapidly broken down by the enzyme phosphodiesterase. An example of hormone operating through adenylate cyclase system is epinephrine that act through the adrenergic beta receptors. Till now, we were talking about the excitatory effect of the receptors. Now, hormones can produce inhibitory effects on a cell, and this may be achieved through the fact that some G proteins such as GI may inhibit the adenylate cyclase, thus inhibiting the formation of CAMP. An example of this mechanism of action is the inhibition of adenylate cyclase through the binding of norepinephrine to the alpha-2 receptors on the presynaptic nerve terminals. Now moving on to inositol triphosphate system. In this system, the hormone receptor G-protein complex interaction triggers the membrane enzyme phospholipase C, which catalyzes the hydrolysis of phosphoinositol PIP2 to two important metabolites: inositol triphosphate, that is IP3, and diacylglycerol. IP3 generates from the endoplasmic endothelium, increase the free calcium, which together with DAG promotes the activation and migration to the membrane of the enzyme protein kinase PKC. PKC may also be mobilized through the entry of calcium into the cell. Now this PKC along with CAM kinase will cause cytoplasmic membrane protein phosphorylation and will ultimately produce the cellular response. Examples of hormones and neurotransmitters which activate this system are epinephrine acting on alpha-1 receptor and acetylcholine on muscarinic cholinergic receptors. These systems are important clinically since they provide substantial number of possible targets for the drugs. Now moving on to kinase-linked receptors. Kinase-linked receptors also use a second messenger system for the signal transduction. They have an extracellular part with hormone binding domain and an intracellular enzymatic catalytic site. The most important cytoplasmic or enzymatic catalytic site have one of four functions: tyrosine kinase activity, tyrosine phosphatase activity, serine or threonine kinase activity, or guanylyl cyclase activity. Depending on this, they are classified into receptor tyrosine kinase, that is RTKase, receptor tyrosine phosphatase, receptor serine threonine kinase, and receptor guanylyl cyclase. When an endogenous ligand, for example hormone in this case, binds to the receptor on the binding site, the nearby receptors couple up to form pairs in order to become an active dimer. This will begin the process of autophosphorylation, where tyrosine residues cross-phosphorylate and become activated as substrate proteins bind to the phosphorylated tyrosines to further relay the signals to the target. This then causes signal transduction and ultimately regulates the gene transcription. So this was all about the membrane receptors. Now switch to the next section to study about the intracellular receptors.